saying, I'll bring my computer tonight to dinner. How's that sound? Right? We can go through pictures then. So we're going to be looking at potatoes that actually came from a commercial storage. This isn't an experiment that I conducted in my laboratory, but a real world setting. I was at the Idaho Potato Consortium October before last in 2017, and I got a phone call from Frelly. And he said, hey Addie, I got to ask you a question. I said, okay. And he said, we got a grower that wants to apply 1,4-DMN so that he can remove all of his harvest bruising. I said, whoa, I've never said that that would happen. I've never ever said that we erased Bruise. Once you've got Bruise, you've got Bruise. Okay, that's just the nature of the game. And after I hung up with him, I got a phone call from Wynn who said, I got a phone call from Frelly who has a grower who wants to use DMN to erase Bruise. And again, Wynn and I were both on the same side on this. We've never said 1,4-DMN does that. But the grower, as you can see, decided to conduct his own test. His potatoes, his shed, and in essence, his money and his time. So, okay, but let's take full advantage of what he's done here. So he did. He went ahead and he treated his shed with 15 parts per million, 1,4-DMN, and we were able to get potatoes prior to treatment and after treatment. So let me get into some of the specifics. The potatoes were harvested between October 13th and October 17th. It took 4 days to clear the fields out and get them into the sheds. On October 24th, Wynn happened to be in the neighborhood, and he collected 4 100-pound bags of untreated tubers. And then those were delivered to the R&D facility on October 26th. This is what I got to look at right here. There were 4 bags, so I took a look at bags 2 and bag 4. And what I wanted to do was just to, let's see, show you here what it looked like just coming right out of the bag and then completely peeled. So untreated potatoes, you can see a little bit of bruising here on the ends, right there, right where it might have hit something or fallen. Right there is a perfect example of a wound with a bruise. And then here's bag 4, a little bit worse for wear, but when you look at the potatoes, they look fine, don't they? They don't look like anything's wrong with them. But you cut into them, and wherever they dropped or got hit, you can start seeing some discoloration there. So you also see some mold starting to form. Okay? So then the commercial storage was actually treated on October 26th. And it received 15 parts per million. And from the treated storage, I received a 100-pound bag of treated potatoes. And then we began to evaluate them. So this is November 6th, and these are untreated potatoes here. And they look pretty good from the outside, nothing disconcerting or upsetting from the view here. But when you cut into them, you can see the color formation here for the bruising on the ends, stem end in particular. You can see some mold formation. So these are not looking that pretty at this point in time. Now here is with 15 parts per million, 1,4-DMN treatment. There is a significant reduction in color formation and in disease formation. Now, I have to admit, I didn't want to believe this. I did not call up Wynn or Frelly or any of the sales guys to come over to my lab and check this out. To me, this was a fluke, and I was going to keep it to myself because this wasn't really happening. So I decided to keep the experiment going a little bit longer. I did draw up some initial conclusions. The potatoes had mechanical damage. I knew for a fact that the day they were harvested, bad weather was coming in, and they were racing against time to get those potatoes into the storage. So they were rough and tumbled in. That I did know. Potatoes were suberizing around the mechanical damage. That's good. That's a healthy sign. Some areas around the mechanical damage had mold formation. That's to be expected. But there is a noticeable difference between the treated and the untreated in terms of bruise color formation. And to my eye, it looked like the treated with the 15 parts per million had less bruise color formation than the untreated. So here we are, November 22nd, and these potatoes are starting to look pretty bad. The color formation is getting darker, the mold is getting larger and more predominant across the surface of the potatoes, and this is with the untreated. Here we have with the treated though. These are essentially quite clean, even where you can see that this potato sustained great impact. Something really hit it hard. There is no color formation there. Same thing over here. I mean, that looked like it just got sliced along something. And again, no color formation. That's when I called up the sales guys. That's when I said they could come over at that point in time. So that I showed them the pictures from the 6th, and I showed them the actual potatoes from the 22nd. I waited a full month until December 21st, so this is 4 weeks after the second set of pictures. And these are untreated potatoes. The difference is really startling here. We've got large areas of mold formation, color formation through here with the untreated potatoes. And here we have the treated. Now we are starting to see some signs of color formation and a little bit of mold, but it is significantly better than the untreated. So at this time I did run out of the untreated control. I didn't have any more to work with, but I did take pictures a month later, so now we're January 29th, and we still have nice white potatoes. There are some areas along here, but I was quite impressed with the difference between treated and untreated potatoes in regards to bruise color formation, and also in regards to the fungal development, the growth. I felt like the fungistatic aspect of 1,4-DMN that I've been hearing about was showing itself here. So my final conclusions was that there was a visible difference in bruise color formation between treated and untreated potatoes. And in particular, the potatoes treated with 15 parts per million 1,4-DMN had less bruise color formation than the untreated control. So have any of you ever seen anything like this before? Yes?
Yep. Last season, but we were treating when they already had dark color, and then after we treated 10 to a 15 ppm, a lot of the dark bruise color was reduced. But the guys that went on door closure within 24 hours or 48 hours versus what they didn't treat, they didn't have any darkening with the bruise. So now they treat 100% on door closure. So when was this, on this example here, how many days after bin fill was this one treated?
Let's see, let me go back and make sure absolutely. They were harvested October 13th to 17th, and then they were treated on the 26th.
Okay.
Yes, treated on the 26th. Okay. So it wasn't an immediate treatment, but about 10 days from the latest potatoes entering the shed. So it's because of this, well, not even experiment, this is an observation. It's because of this observation that one of the largest projects that I've ever been a part of has come to 1/4 group. So let me zip through this here. So untreated, treated, untreated, treated. So do I just move into the next one?
No, it's on a separate separate PowerPoint.
So there's two growers here at the show from Manitoba. So that was last year on fresh yellows and reds. This year I treated probably about 3 million bags of more than the season before. Now the guys are doing the processing potatoes.
Yeah.
Now, there's growers here with me that these are non-believers, and I got them to try it this year on each bin, and they're seeing a 20 to 15% increase on their bruise-free hauling to McCain's. McCain's finally said, yeah, we're starting to see a benefit, but we got more issues than that with the frozen potatoes.
Mm-hmm.
There's guys here to show that they've never, in all their farming, they've never seen as high a bruise-free on their potatoes.
Well, Wynn can talk to that with some of the other projects that we've been working on as well.
But your success has been with the early shots.
I've done everyone early. Now I'm going to be doing one next week at 15 ppm. We're doing pulling samples, bringing them to Ag World, doing a bruise before, and then I want to do a test 3 weeks, 4 weeks, and 5 weeks and see the bruise difference if there is one. But these are all done cold, so they're going to be.
Yeah, should have some if they didn't.
They're going to grow dormancy already. Now the cold ones are starting to sprout.
Yeah. Okay. All right.
So now I'm going to talk about, like I said, one of the largest projects that I've ever been a part of. And this is so exciting for me because at 1,4 Group, as a research scientist for a chemical company, I have to be honest, my work is scrutinized because there's an assumed bias. They assume that I am going to be biased for 1,4-DMN, regardless of all the measures that I take. And I even go so far as to try to be, ultra pure in all of my research. There'll still be that assumed bias that I am leaning towards the chemical that in essence signs my paycheck. I get it, I really, really do. Which is why I want to work with universities where there is more of a non-biased approach to science. And I'm so honored to be able to work with Dr. Michael Campbell from Penn State and also Dr. Michael Thornton from the University of Idaho. And I'm sure most of you have heard of Dr. Michael Thornton as well, right? So, let's see, a little bit of a background here. 1,4-DMN is registered by the EPA as a biocontrol. That means that the 1,4-DMN, the 1,4-dimethylnaphthalene, that's the active ingredient in 1,4-site and 1,4-C, is found naturally in the potato. But it can't be called organic. There are not sufficient quantities of that naturally occurring DMN to supply all of our needs. So we have to go to a factory and synthesize it. That's why it's called a biocontrol. It has the biological aspect of being found naturally in natural systems in the world, but we have to make it synthetically in a factory. So that's why we're in that special category. It is still a gentler chemistry than a conventional chemistry. Conventional chemistries are usually man-made, and they tend to be sides, meaning kill. Bacterial side, it'll kill bacteria. Fungicide, it'll kill fungus. Being a biocontrol, we're fungistatic. This is actually very good news for us when you are talking to your customers because they want gentler chemistries. They want the same effectiveness as conventionals, of course, but they want everything to be gentler. How many people are you working with that have sustainability mission statements? We want to be sustainable. We want to be green, but we want it to work, right? So I urge all of you actually to learn the language of what biocontrols mean. These buzzwords are becoming more and more important to your customers and to their customers. So through the years, secondary benefits of 1,4-DMN have been noted. Not just the sprouting, but growers have noted that there's a fungistatic effect, a microbial static effect, that there's a reduction in weight loss, that there's an ability to control respiration of the potatoes when they're stressed. There's a whole host of secondary benefits. So Dr. Michael Campbell's gone through his research, and he's been able to demonstrate a genetic response within the potato following a 1,4-DMN application. 1,4-DMN is found naturally in the potato, so applying it externally would, I would think, follow naturally that certain genes would be turned off and on as if it was innately found in the potato. Osmotin proteins are produced. Some of them are associated with water holding. Hold on to that water, don't let it go, and that's associated with a reduction in weight loss. Sprout inhibiting proteins, which we're all very knowledgeable about, and then PR proteins associated with an increased immune response, maybe against pathogen attack. Again, 1,4-DMN, the benefits, we have seen it in large-scale commercial storages around the world. I mean, 1,4-DMN is now in Europe, it's in New Zealand. We've seen its effect in some of the storages even in the Middle East. So we are seeing 1,4-DMN works consistently around the world. It's worked in all of my research storage capacities. I have jars, ammunition cans, barrels, and research bins. And also in Dr. Michael Campbell's small-scale research chambers. However, there hasn't been a direct link between the genetics and the physiology. And this came to light when we were asked to write an article for Potato Pro. And our article was rejected, and I was shocked and aghast that it was rejected. And the reason why is that they said that there wasn't a strong link connecting what happens genetically with what happens physiologically. Where I do believe a geneticist would say, well, wait a minute, you've made a protein, that's a physical response. But the potato physiologists want to see a response in the field or in the storage, you know, in grand mass, more than just a small protein. So I'm like, okay. So what we decided to do was see if we could get a geneticist, Dr. Michael Campbell, together with Dr. Michael Thornton, who's a potato physiologist. And I call it Mike and Mike. They met and they work well together. There's no politics involved in it, because sometimes the potato world can be quite filled with politics. And instead, what we're able to do is to bring two scientists together to work on a common goal. And this is actually pretty large in the research area, to have a geneticist cross over and work with a physiologist. I mean, even at the PAA meeting, they have separate rooms for their meetings. They just go separately. But here we have two guys coming together. So the purpose of this entire project, to determine if the physiological response of potatoes to DMN, noted at the genetic and cellular level, corresponds to a physiological response involved with sprouting, disease resistance, weight loss, and bruising on potatoes grown and harvested from University of Idaho Palmer Research Station. Okay. So the potatoes are going to be, well, we've already grown them, but the potatoes were grown and harvested at U of I Research Station in Parma under the watchful eye of Dr. Michael Thornton. Those potatoes were brought to the 1,4 Group Research Center, and that's where I come in. The potatoes are stored in my facility there, and I do the treatments. But because of the assumed bias, that's where I step back. After that, Dr. Campbell and Dr. Thornton do their thing. Thornton comes in and he does all of the bruise evaluation. He does all of the disease evaluation, and he does the weight loss evaluation. And you'll see that coming up. Dr. Campbell came in for 1 full week, and he collected samples for his future DNA and RNA analysis. And then he trained my chemist, Art Espinosa, to do those same tests later on in the year. So you can see that I've removed myself from the project. I simply store the potatoes there and I treat them there. And I've done this on purpose so that the physiologist and the geneticist can be the ones that are evaluated. So we had russet Norcota, Clearwater russet, and russet Burbank seed. They were cut and planted at U of I Research Station in April. And my team went out every week and we took care of the fields. We made sure they're properly watered, fertilized, cared for. We did that all summer. And then we harvested them week of September 24th. All 3 varieties were windrowed, but then hand harvested into bags and then brought into the 1,4 Group Research Center. We stored them for 1 week at 10 degrees C, which is 50 degrees Fahrenheit. And so the bruising of the potatoes is going to take place inside of my shop, and all chemical treatments is going to take place outside of my shop. That way the chemical wasn't going to waft or move on to any of the untreated potatoes. So that's what this is saying. And from the mesh bags, the potatoes were either gently loaded into the barrels, if that's what the experiment called for, or hand bruised and then put into the barrels. So here's the treatment protocol. This is one of the machines that I used here. And I apply DMN between 550 and 580 Fahrenheit. Let's see, I don't know if I have to go into this much detail, except that I use 15 parts per million, or 4 mils of 1,4-DMN to achieve the level of 1,4-DMN residue that I wanted. And let's see, then at 2 days after treatment, I collected 1,4-DMN residues. So here's the very first experiment that we set up. Fusarium dry rot control. We chose Clearwater russet. I've heard that Clearwater russet is having big challenges out there with Fusarium. Have all of y'all heard that too? Yeah. So Clearwater russet, we brought them into the lab, and then we had a table, and we drew a 12-inch line right about here. We brought the stem end to that 12-inch line and dropped it on a steel plate, flipped it, and dropped it. And we did that to 1,500 pounds of clear water. We had, let's see, 3 barrels here that were untreated, and then 3 barrels that were treated with 15 parts per million 1,4-DMN, 7 to 10 days after harvest. That's what DAH means. And then retreat at blinking stage if necessary with monthly evaluations. Now what these red stars indicate are sites where Dr. Campbell is sampling for the genetic response. And so you can see that we're looking at before treatment and after treatment. Next we have the weight loss study, and we chose russet Burbank for the weight loss. Burbank potatoes were placed into the research barrels. Again, a 15 part per million treatment with DMN on one side. They're going to be weighed weekly, and then after 1 month, weighed monthly until the end of season. Same thing with the untreated over here.
So
Next we have
Campbell,
pressure.
question.
Yeah.
So, Mike, when you go and do these samples at these stars where you're going to do that, then what does that mean for us in here? What are you seeing or comparing before and after? Are you talking about transcripts or?
Yeah, gene expression, transcripts, RNA that goes up and down. We're looking at genes that go up and down at each one of those points. So the question would be, how do you want to set up the comparison? And probably on an experiment like this, you'd want to compare the genes associated with treated with DMN as opposed to treated with CIPC, right? Because then you're going to be looking at genes that go up in response to DMN compared to CIPC. Okay.
So now we're going into pressure bruising. Within my research lab, I have, over here, thank you. So within my laboratory, I am fortunate enough to have someone who developed a barrel in which even pressure is pressuring down on top of the potatoes, and I can mimic an 8-foot pile or a 20-foot pile of potatoes. And so what we did here, we used russet Dakota, and we did not bruise them because they were going to be pressure bruised. And we divided into 4 barrels that were treated, and then 4 barrels that were also treated over here with CIPC. Now this was Dr. Michael Thornton's suggestion to treat with CIPC rather than untreated entirely. He was worried that if the potatoes sprouted, then they would succumb to pressure bruise anyway because the moisture from the potato would go into the sprout. So I said, okay, so I've got 4 barrels with DMN and 4 barrels with CIPC. Now what we have noted out in the field is that 5 to 7 days before unloading, if you treat with the 1,4-DMN 5 to 7 days before unloading, there's an apparent reduction in bruise color formation. So based upon what I've learned from applicators and growers out in the field, I decided to apply 1,4-DMN again after 4 months, which will be end of January, 1st of February. and then evaluate after 1 week of sitting out at room temperature. The untreated control over here, I call untreated because it has CIPC, it doesn't have the DMN. So it receives that 1 CIPC treatment in the fall, and then we'll evaluate at the same time that the DMN treated barrels are evaluated. Now we have harvest bruising. This is supposed to mimic when the potatoes have been pretty banged up coming out of the field. And maybe you're chasing bad weather, maybe you didn't have the speeds correct on the harvester, who knows what, right? But they got banged up coming out of the field. So again, we used russet Norcota. We treated, I got 3 different sections here. First one here is treat with 15 parts per million, 1,4-DMN, 7 to 10 days after harvest. And then treat again 5 to 7 days before unloading, and then evaluate. This second set right here does not receive the early treatment, only the late treatment of DMN, and then evaluated. And then these here are completely untreated. So this is early treatment, late treatment, evaluate. This one is late treatment only, then evaluate. And this one is untreated all the way through. And then finally, I've got non-bruised potatoes. These are unloading from the shed. You had a great harvest, you got them in with minimal bruising, they've been sitting in storage for a while, and now you're pulling them out, and they're starting to receive bruise at that point from the unloading process. So these were gently harvested, and they were not bruised whatsoever. And when we put them into the barrel, so it received an early treatment of DMN here, 7 to 10 days after harvest. And then at 2 days after treatment, then they were dropped, hand dropped to have the bruise formation. So these were chemically treated, then bruised. With the other slide, they were bruised and then chemically treated. So we're trying to see every aspect of bruising we can think of. These right here remained untreated, but then we bruised them, and then we'll evaluate. And then these have had no treatment whatsoever on them at all. Again, you can see the red stars where Dr. Campbell is sampling. So we're trying to take samples for the genetic response prior to treatment, after treatment, prior to bruise, after bruise, prior to bruising, then treatment, prior to treatment, then bruising. You can see that our grid is getting massive with all of the samples that we're trying to do here. Next steps right here. Let's see, so I'm expected to pull the pressure barrels and the harvest bruising out the week of January 29th. And it'll take us about 3 weeks to evaluate everything at that point. The fusarium I've been looking at monthly, and we'll continue to evaluate through that probably until May or June. And the genetic results can take a lot of time, and I'm just saying many months if they're right now. So this is what we're working on right now. It's, for me, it's, I guess, one of the most powerful projects I've ever worked on. It's extremely intricate. How many samples have you taken, Dr. Campbell? Hundreds. Hundreds of samples. And we're not even done yet. We're simply not even done yet. Oh, yes. This is my last slide, but this is where dinner's going to be tonight. 6 p.m. at the Moonshine Grill, 303 Red River Street.
That's just on the back side of the convention center, back on this this corner, I believe, if I'm not turned around.
So we do have an NDA, a non-disclosure agreement, between Penn State, University of Idaho, and 1,4 Group. Whereas I'm allowed to show you the structure of the experiment, I'm not allowed to display results. I have to turn in the information to an independent statistician first. Again, that removes me from bias. Like for the weight loss, those potatoes are down in Dr. Thornton's laboratory. He is the one weighing them. They're completely under his control. I greatly prefer this. When we go to publish, I hope just to be, I don't know, maybe a supporting author so that there's no question about the authenticity of this study. So, do you guys have any questions at all?
I get a lot of scrutiny just because you funded my work.
Oh,
So I have to usually justify to editors of journals and even to my university that I have a vested interest. I'm not an investor in the 1/4 group. I'm not making money from this research, that kind of stuff. So I get a lot of scrutiny too. So I mean, it should be. Right?
yes, yes.
But
Well,
it is that way.
yeah, it is what it is. So, well, I can understand it. There's been some companies that have been rather free sometimes with their evaluations. So, anyway, I do expect to have results. Well, probably not from the genetic side of it. I think the physiological side will come out sooner.
So,
And what could estimated time be to find out at the end of it all the results? When would that timeline be?
This coming fall, maybe?
Yeah, probably
It's
around
gonna be a while.
fall.
Okay.
The physiological results are going to be a lot easier to display, but then there's also the connection of the physiological results to the genetic results. And Campbell and I were talking about it this morning. The number of papers that could come out from this is enormous.
Yeah.
Any other questions for Addie today? Listen, we appreciate you guys coming out today. Some of you guys have seen this too in 3 times, Peterson, so thanks for staying awake. Others of you have not seen some of this information before from Dr. Campbell and Dr. Waxman, so we appreciate your time coming in today and sitting here and learning more about what Dr. Campbell's experiencing and seeing. We hope it'll help you as you go out in the field, be a good sales tool. Dr. Campbell and Addie will be here for a little while, so if you guys have any one-on-one questions or continued conversations with Dr. Campbell or Addie, we have the room this afternoon. You're all invited, spouses, everybody. Come to dinner tonight at the Moonshine, kind of a Southern comfort type food tonight. So we'd like to have you there. Continue the evening. We appreciate it. Thank you very much.
Thank you, guys.